Let's review the functions of coagulation factors. The primary substrate is factor I, also called fibrinogen. Fibrinogen is converted into fibrin, which forms the structural basis of the clot. Cofactors accelerate enzymatic reactions in the clotting cascade. They include factor III, factor V, and factor VIII. In addition, high molecular weight kininogen, also called HMWK, acts as a cofactor. Most clotting enzymes belong to a group known as serine proteases. These enzymes cleave peptide bonds to activate downstream factors. The serine proteases include factor II, factor VII, factor IX, factor X, factor XI, and factor XII. There is also a single transamidase enzyme, factor XIII, which stabilizes the clot by cross-linking fibrin strands. The bottom line? Coagulation factors work together in distinct roles. Some serve as substrates, some as cofactors, and others as enzymes. Each role is essential for effective clot formation and stabilization. Let's walk through the characteristics of coagulation protein groups. First, we have the contact group. This group includes factor XI, factor XII, prekallikrein, and high molecular weight kininogen. These proteins are not vitamin K dependent. They are also not consumed during the clotting process, meaning they remain in plasma after clotting occurs. Next, we have the prothrombin group. This group contains factor II, factor VII, factor IX, and factor X. They are vitamin K dependent. Most of them are not consumed during clotting, with the exception of factor II, also known as prothrombin, which is consumed when it's converted into thrombin. Finally, we come to the fibrinogen group. This includes factor I, factor V, factor VIII, and factor XIII. These proteins are not vitamin K dependent. However, they are consumed during clotting. For example, fibrinogen is converted into fibrin, factor V and factor VIII are used up as cofactors, and factor XIII stabilizes the clot through cross-linking. The bottom line: the contact group helps initiate coagulation but remains in circulation. The prothrombin group requires vitamin K for synthesis and includes most of the vitamin K-dependent factors. The fibrinogen group is unique because its members are actually consumed during clot formation. Let's review factor names and clot regulation. Factor names. Coagulation factors are generally noted by Roman numerals. There are a few exceptions. Prekallikrein and high molecular weight kininogen, which are written out by name rather than number. Stabilized cross-linked fibrin. How do we turn clotting on? The clotting cascade can be activated in two ways. Intrinsically, when collagen is exposed, it activates factor XII in the contact system. Extrinsically, when the endothelial cell membrane is disrupted, tissue factor, also called tissue thromboplastin, forms a complex with factor VII. This complex directly activates factor X. Cofactors in action calcium helps factor VIII and activated factor IX bind to the platelet phospholipid membrane. Together, these cofactors activate factor X. In the common pathway, factor V serves as the cofactor for activated factor X, again requiring calcium and phospholipid. This arrangement forms the prothrombinous complex, which converts prothrombin into active thrombin. Thrombin is a key enzyme in clotting. It cleaves small peptides from fibrinogen, producing fibrin monomers that polymerize into insoluble fibrin strands. Thrombin also activates factor XIII, which cross-links fibrin strands at the D region. This is the origin of the D-dimer fragment detected in laboratory tests. Turning it off, the coagulation process must be carefully controlled. Heparan sulfate, located on endothelial cells, binds antithrombin. This interaction inactivates many of the serine proteases. Heparin therapy works in the same way. Additionally, activated protein C, together with its cofactor protein S, inactivates factors 8 and 5. This requires binding to thrombomodulin, the receptor activator complex on the endothelial surface. The bottom line: coagulation is a balance. It can be turned on by intrinsic or extrinsic pathways, sustained by cofactors and thrombin, and turned off by natural anticoagulants such as antithrombin and the protein C protein S system. Now let's explore the fibrinolytic system, which regulates the breakdown of clots. Turning it on. The fibrinolytic system can be activated in two ways. Intrinsically, through collagen exposure via the factor XII contact pathway, which also initiates intrinsic clotting. Extrinsically, through tissue plasminogen activator, or TPA. These activators convert the precursor plasminogen into the active enzyme plasmin. What does it do? Plasmin is the key enzyme of fibrinolysis. It cleaves fibrin strands into soluble fragments known as fibrin degradation products. These fragments are identified as X, Y, D, and E. Fibrin degradation products can come from two sources, either from the breakdown of a fibrin clot, called fibrinolysis, or from the breakdown of unclotted fibrinogen, called fibrinogenolysis. The D-dimer fragment, which is clinically important, is formed only from the breakdown of a cross-linked clot. That makes D-dimer specific for clot degradation. Turning it off. The fibrinolytic system must be controlled to prevent excessive bleeding. Tissue plasminogen activator, or TPA, is inactivated by tissue plasminogen activator inhibitor, known as TPAI. This prevents overactivation of plasminogen. If active plasmin escapes from the clot site, it is rapidly bound and inactivated by alpha-2 plasmin inhibitor. This prevents uncontrolled degradation of circulating fibrinogen. The bottom line: the fibrinolytic system is a balance. It is turned on by activators that generate plasmin. It breaks down fibrin into soluble fragments, producing D-dimer from cross-linked clots. It is turned off by inhibitors that neutralize TPA and plasmin. This ensures clot removal happens only where and when it is needed. Now let's talk about excessive and inappropriate fibrinolysis. One of the major conditions where this occurs is disseminated intravascular coagulation, or DIC. In DIC, the body responds to excessive clotting by turning on fibrinolysis inappropriately. This becomes a hallmark feature of the disorder. Another setting is liver disease. The liver normally clears fibrinolytic activators and produces inhibitors. When the liver is diseased, these activators are not properly cleared, and inhibitors are reduced. The result is unregulated fibrinolysis. Finally, excessive fibrinolysis can occur with cancer or after surgery of the prostate or urinary tract. In these situations, urokinase, an enzyme capable of activating plasminogen, can leak into the circulation. This leakage leads to inappropriate activation of fibrinolysis. The bottom line: inappropriate fibrinolysis can be triggered in three main contexts: excessive clotting, as in DIC, impaired regulation, as in liver disease, or direct urokinase leakage, as seen in prostate or urinary tract conditions. Each pathway ultimately causes the same issue: an imbalance where clots are broken down too aggressively. Now let's review specimen collection and handling for coagulation testing. The anticoagulant of choice is sodium citrate at 3.2% concentration. This works by binding calcium. Since calcium is essential for both coagulation and platelet aggregation studies, this prevents premature clotting in the tube. For accuracy, the ratio of whole blood to anticoagulant must be 9 parts blood to 1 part citrate. If this ratio is not correct, test results may be inaccurate. Specimens should be collected in plastic tubes or siliconized glassware. Regular glassware should not be used because plain glass activates factor XII and platelets may adhere to the surface, altering results. In the past, coagulation samples were often collected after other tubes to avoid tissue thromboplastin contamination. However, with the use of modern evacuated tubes, or vacutainers, Coagulation samples can now safely be drawn first. Historical concerns about contamination are no longer supported. Sample quality is also important. Hemolyzed samples should not be used for platelet aggregation studies because red cells release ADP, which interferes with results. Lipemic samples can also cause problems. Excess lipids may obscure changes in optical density, leading to inaccurate measurements in both coagulation and aggregation studies. The bottom line? Proper anticoagulant concentration, correct blood-to-citrate ratio, appropriate collection tubes, and avoiding hemolyzed or lipemic samples are all critical for reliable coagulation and platelet aggregation testing.